Hello friend and welcome to Facts for Sleep. Tonight we're going to explore the deep forest. The forest that exists far from roads and cities. The forest where light arrives only in small pieces and where the ground is always soft and damp. If you find this kind of slow exploration helpful, you may want to subscribe. There is much to discover here, and all of it is real. So find a comfortable position. Let your breathing slow and let the forest come to you. A deep forest begins with darkness. Not complete darkness, but a kind of filtered dimness. When sunlight reaches the top of a tall forest canopy, it must pass through many layers before it reaches the ground. The upper canopy of a mature forest can be 30 meters above the ground or more. By the time light filters through the leaves at that height, it has already lost much of its energy. Then it must pass through a second layer of smaller trees growing beneath the canopy, then through a layer of shrubs and ferns, then through low mosses and ground cover. By the time any light reaches the forest floor, it may contain only 1 or 2% of the original sunlight that fell on the treetops. The rest has been absorbed by the leaves above. This low light changes everything. It determines which plants can grow on the forest floor. It determines which animals can find food there. It shapes the temperature of the air close to the ground. Because so little energy reaches the forest floor, that layer stays cool even in summer. In a dense forest on a warm day, the temperature at ground level can be several degrees lower than the temperature in an open meadow nearby. The trees above act as a kind of thermal shield. They absorb the heat of the sun and release it slowly and gently through the night. The first thing you notice when you enter a deep forest is the sound, or rather the quality of the sound. Sound behaves differently among trees. The trunks and branches absorb and scatter acoustic energy. High-pitched sounds are broken up quickly, low sounds travel further. The result is a kind of natural acoustic filtering. The forest removes sharp and harsh frequencies from the soundscape. What remains tends to be soft, the low creak of branches, the distant drip of water, the muffled shuffle of a small animal in the undergrowth. Many researchers have noted that the acoustic environment of a deep forest has properties that are measurably different from open environments. Sound is both quieter and more even in its distribution. The ground beneath a deep forest is not simply soil. It is a layered system that has been building for decades or centuries. The topmost layer is called the litter layer. It consists of freshly fallen leaves, broken twigs, fragments of bark, dead insects, and other recently deposited organic material. Just below this is a layer called the duff. Here the material is more decomposed. Individual leaves are no longer recognizable. The organic matter has broken down into fine dark particles. Below the duff is the humus layer. This is where the decomposed organic material has fully merged with the mineral soil beneath it. Together these layers form what is sometimes called the organic horizon of the forest soil. In a mature forest, this organic layer can be 20 centimeters deep or more. It took many generations of trees to create it. This layered soil is extraordinarily alive. 1 teaspoon of healthy forest soil contains more living organisms than there are people on the planet. Most of these organisms are invisible to the naked eye. Bacteria, archaea, single-celled fungi and protozoa make up the majority. But there are also larger organisms present. Nematodes, which are tiny roundworms, exist in almost every forest soil sample. Mites and springtails occupy the spaces between soil particles. Earthworms create channels through their deeper layers. Beetle larvae tunnel through this duff, and threading through all of it are the hyphae of fungi. These thin white threads extend for enormous distances through the soil. A single teaspoon of forest soil may contain several kilometres of fungal threads. Fungi are perhaps the most important organisms in the deep forest that most people never see. They exist in two main forms. The first is the form most people recognise. The mushroom is simply the fruiting body of a fungus. It is the reproductive structure that appears above ground to release spores, but the main body of the fungus lives entirely underground. This underground portion is called the mycelium. It consists of those branching threads called hyphae. The mycelium of a single fungal organism can spread through hundreds of square meters of forest soil. Some fungal organisms extend over many hectares. One fungal organism in a forest in Oregon in the United States has been measured at nearly 10 square kilometers. It is among the largest living organisms ever recorded. Most of the fungi in a forest floor are not simply decomposers, they form partnerships with trees. These partnerships are called mycorrhizal associations. The word mycorrhiza comes from the Greek words for fungus and root. In a mycorrhizal association, the fungal hyphae wrap around or grow into the fine root tips of trees. The two organisms exchange materials in a way that benefits both. The fungus provides the tree with water and dissolved minerals, particularly phosphorus and nitrogen, which the fungal threads can reach far more effectively than the tree roots alone. In return, the tree provides the fungus with sugars. These sugars are produced by photosynthesis in the leaves high above. The tree sends a portion of its sugar production down through its roots and into the fungal network below. This exchange happens continuously. A mature tree may send between 10 and 40% of all the sugars it produces via photosynthesis directly into its mycorrhizal fungal partners. This is not a small contribution. The tree is investing a significant portion of its energy into maintaining this underground partnership. In return, the fungal network effectively extends the reach of the tree's root system enormously. The hyphae can penetrate spaces too small for roots. They can access phosphorus locked in mineral particles that roots alone could never extract. Because of this exchange, trees growing with mycorrhizal fungi are measurably healthier, taller, and more resistant to drought and disease than trees grown in soil without them. The same fungal networks that connect individual trees to their fungal partners also connect multiple trees to each other. When two trees are both connected to the same fungal network, carbon can move between them. Researchers have demonstrated this using radioactive carbon tracers. When a tree is labelled with a radioactive form of carbon and then monitored, that labelled carbon can later be detected in neighbouring trees that share the same fungal network. This means that the forest floor is not just a place where individual trees send nutrients down, it is a place where nutrients actively move between trees through a shared underground system. This discovery has changed how many forest ecologists think about the forest. Rather than seeing a forest as a collection of individual competing trees, some researchers now describe it as a kind of superorganism, or at least as a community with a degree of connectivity that not previously understood. The flow of carbon through fungal networks can be substantial. Seedlings growing in the shade beneath tall trees receive more carbon than they could produce themselves through photosynthesis. Some of this carbon appears to come through the fungal network from the larger trees nearby. Whether this represents something like intentional support is debated. But the physical flow of material is well documented. The chemistry of forest soil is shaped by more than just fungi and roots. Leaves falling from the canopy carry chemical compounds with them. As these leaves decompose, those compounds enter the soil. Different tree species produce leaves with different chemical profiles. Oak leaves are rich in tannins. Beech leaves decompose slowly because they contain compounds that resist breakdown. Pine needles are acidic and can lower the pH of the soil beneath pine forests over time. The soil under a beech forest smells and looks different from the soil under a pine forest or an oak forest. Each tree species shapes its own soil environment over centuries of leaf fall and root activity. The roots of trees also release chemicals directly into the soil while they are still alive. These released compounds are called root exudates. A tree produces a continuous flow of sugars, amino acids, organic acids, and other compounds from its living root tips. These exudates attract specific bacteria and fungi to the area immediately surrounding the roots. This zone of chemical activity around roots is called the rhizosphere. The rhizosphere contains a microbial community quite different from the general soil microbial community just a few centimeters away. The tree is actively cultivating its own community of soil organisms through the chemistry it releases from its roots. Bacteria in the rhizosphere break down organic matter and release nitrogen and phosphorus in forms the tree can absorb. Some bacteria fix nitrogen from the air, converting it into ammonia that becomes available as a plant nutrient. These nitrogen-fixing bacteria are particularly important in forests where nitrogen is limited. Older trees, which often grow along forest streams, have a special relationship with nitrogen-fixing bacteria in nodules on their roots. The bacteria inside these nodules can fix large quantities of atmospheric nitrogen. This makes older trees a significant source of new nitrogen in forest ecosystems. Forests containing older trees tend to have soils with higher nitrogen levels than forests without them. Water moves through a forest in ways that are different from water moving across open land. When rain falls on a forest canopy, much of it is intercepted by leaves and branches before it ever reaches the ground. Some of this intercepted water evaporates directly from leaf surfaces back into the atmosphere without ever touching the soil. The rest flows along branches and down trunks, a process called stem flow, or it drips from leaf tips in a process called throughfall. These processes slow the movement of water. Rather than falling directly onto the soil surface, the rain arrives gradually and gently, distributed across the forest floor in a gentler manner than it would fall on open ground. When water does reach the forest floor, the organic litter layer absorbs it like a sponge. The duff and the humuslet below can hold enormous quantities of water. A well-developed forest floor organic layer can absorb and retain many times its own weight of water. This water is held in the spaces between soil particles and in the tissues of living organisms. It moves slowly downward through the soil profile rather than running off across the surface. This means that a forest acts as a natural water regulator. It slows the movement of rainwater through a landscape. After heavy rain, a forested watershed releases water gradually through streams and rivers over days and weeks. A deforested watershed releases the same rain rapidly in floods that subside just as quickly. The trees themselves are major movers of water. Through their leaves, trees release water vapor in a process called transpiration. Water is absorbed from the soil by roots, moves up through the trunk in tubes called xylem vessels, and exits through tiny pores in leaves called stomata. A mature, deciduous tree can transpire hundreds of litres of water on a single warm day. A large forest transpires enormous quantities of water. This water enters the atmosphere above the forest and contributes to cloud formation. Some research suggests that forests generate a significant portion of their own rainfall through this process. The trees pull water from the ground, release it into the air, and the air above the forest becomes humid enough to produce rain that falls back onto the forest. The movement of water up through a tree is itself a remarkable physical phenomenon. There is no pump. The heart of a tree does not beat. Water moves upward through the xylem because of a process driven partly by the evaporation at the leaf surface and partly by the cohesive properties of water molecules. Water molecules are attracted to each other. When a water molecule evaporates from a leaf surface, it pulls the next water molecule up slightly. That molecule pulls the one below it. The result is a continuous column of water pulled upward through the tree by the tension created at the top. In a tall tree, this means water has been held under significant negative pressure. In a sense, the water in the upper parts of a tall tree is under tension rather than compression. It is being pulled upward rather than pushed. This system is called the cohesion-attention mechanism, and it is the primary explanation for how water reaches the tops of trees 100 meters tall. The bark of a forest tree is its outermost layer, and it serves as both a physical shield and a complex chemical factory. The outermost bark, called the ritidome, is dead tissue. It consists of flattened dead cells that protect the living wood beneath from physical damage, temperature extremes, pathogens, and insects. The texture and pattern of bark varies enormously between species. The deeply furrowed bark of an old oak is quite different from the smooth pale bark of a young beech. These differences are partly genetic and partly the result of how the tree grows. As the tree grows outward, the bark cracks and folds in patterns determined by the growth rate of the wood beneath it. Oak grows in irregular bursts, producing deep, irregular furrows. Beech grows more steadily, and its bark stretches rather than cracking, remaining smooth even as the tree ages. Just beneath the outer bark lies a thin and critically important layer called the phloem. The phloem transports sugars produced by photosynthesis in the leaves down through the tree to the roots and to any other part of the plant that needs them. If you strip the bark from a tree in a ring all the way around its trunk, you sever the phloem. The roots are then cut off from their sugar supply, and the tree will die, even if its leaves continue photosynthesizing for a while, and even if its water-conducting xylem is intact. This is called girdling, and it has been used both intentionally and accidentally to kill trees. Animals that strip bark in rings can girdle and kill trees. Certain fungi that grow beneath the bark can achieve the same result by destroying the phloem tissue. Between the phloem and the xylem lies the vascular cambium. There is a single layer of actively dividing cells that is responsible for the growth of the tree trunk in width. Every year the cambium divides to produce new xylem cells on its inner surface and new phloem cells on its outer surface. The xylem produced in spring when water is plentiful and growth is fast consists of large cells with thin walls. The xylem produced in late summer when growth slows consists of smaller cells with thicker walls. The difference in density between spring wood and summer wood creates the visible rings we call annual growth rings. Each ring represents 1 year of growth. By counting the rings of a tree stump, you can determine the age of the tree. By measuring the width of each ring, you can read a history of growing conditions. Wide rings represent good years with ample rain and warmth. Narrow rings represent difficult years with drought or cold. Old trees in a deep forest carry decades or centuries of this history in their wood. A tree that began growing 300 years ago has experienced every drought, every cold year, every insect outbreak that affected that forest during that time. The record is preserved in the wood. Dendrochronology, the science of breeding tree rings, has been used to reconstruct climate records going back thousands of years by matching ring patterns from living trees with those from ancient preserved wood. Ring patterns from different trees in the same region tend to match because they all experience the same weather. But overlapping these patterns, scientists have assembled continuous ring records, extending back more than 10,000 years. The interior of a living tree is not entirely alive. The outermost rings of xylem, typically the most recently formed, make up what is called the sap wood. This wood still contains living cells called ray cells and parenchyma cells distributed among the dead xylem vessels. These living cells perform metabolic functions including storing starch and defending against pathogens. The inner rings of older wood make up what is called the heartwood. In most species, the heartwood is no longer physiologically active. Its cells are dead. But the heartwood is not simply dead wood. The tree deposits chemical compounds into the heartwood over time. These compounds are called extractives, and they include tannins, resins, oils, and various antimicrobial substances. These extractives give heartwood its darker colour and its resistance to rot and insect damage. The heartwood of a tree is in a sense a long-term chemical investment. It becomes more valuable as a structural material as it ages. The canopy of a deep forest is a world of its own. Up among the branches where light is abundant and the temperature fluctuates more than it does at ground level, there is a distinct community of organisms that rarely descends to the ground. Lichens coat the upper surfaces of branches. Mosses grow in the forks where water collects. Epiphytic plants in tropical forests grow directly on branches without touching the ground at all. Birds occupy the canopy and rarely venture below the understory. Insects specific to the canopy environment spend their entire lives in the treetops. Until the development of canopy access techniques in the late 20th century, this world was almost entirely unstudied. Researchers who first climbed into old-growth forest canopies using ropes and platforms discovered species that had never been documented before. The leaves that form this canopy are themselves remarkable structures. A leaf is essentially a solar panel combined with a gas exchange system. The outer surface of a leaf is covered by a waxy layer called the cuticle that reduces water loss. Just beneath the upper surface is a layer of elongated cells called the palisade mesophyll. These cells are densely packed with chloroplasts, the organelles that contain the green pigment chlorophyll and carry out the light-capturing reactions of photosynthesis. Below the palisade layer is the spongy mesophyll, a loosely arranged layer of cells with large air spaces between them. These air spaces connect to the outside through the stomata, the small pores on the leaf surface. Carbon dioxide enters through the stomata and oxygen exits. Water vapor also exits through the stomata during transpiration. A single leaf may contain more than 30 million chloroplasts. Each chloroplast contains stacks of membrane structures called thylakoids, where the light-dependent reactions of photosynthesis occur. Light energy strikes molecules of chlorophyll and energizes electrons. These energized electrons are passed along a chain of proteins embedded in the thylakoid membrane. As they pass along this chain, they drive the production of a molecule called ATP, which is the main energy currency of the cell. The same process also generates a molecule called NADPHSOS, which carries electrons for use in the next stage of photosynthesis. At the end of the electron transport chain, the electrons are replaced by electrons stripped from water molecules. This splitting of water releases oxygen as a byproduct. The oxygen produced by photosynthesis in the leaves of forest trees is the same oxygen that fills the air above a forest and that we breathe. The second stage of photosynthesis uses the ATP and NADPH produced in the light reactions to convert carbon dioxide from the air into sugar. This process is called the Calvin cycle, and it occurs in the fluid-filled space inside the chloroplast called the stromy. Carbon dioxide molecules are captured by an enzyme called rubisco and attached to a 5-carbon molecule called ribulose bisphosphate. The resulting 6-carbon molecule immediately splits into 2 3-carbon molecules. These are then processed in a series of reactions using the ATP and NADPH from the light reactions until eventually a 3-carbon sugar called glyceraldehyde 3-phosphate is produced. This sugar can be used to build glucose, starch, cellulose, and all the other carbon-containing compounds the plant needs. The air in a deep forest has a chemical composition slightly different from the air in an open field or a city. Trees and other forest plants release a variety of volatile organic compounds through their leaves. The most commonly discussed of these are the terpenes, a particular class called monoterpenes. These are the compounds responsible for the characteristic smell of a pine forest or a eucalyptus grove. But all forests release terpenes in varying quantities. These compounds enter the atmosphere and can undergo chemical reactions with other compounds in the air, contributing to the formation of tiny particles. These particles scatter light, which is one reason why forested mountains often have a slightly blue haze above them. The Great Smoky Mountains in the eastern United States are named for this effect. Some researchers examined whether the chemical compounds released by forests have biological effects on people who breathe them. Studies conducted in forests in Japan as part of a practice called Shinrin-yoku, or forest bathing, have measured physiological responses in people walking slowly through forests compared with people walking in urban environments. Some studies reported lower levels of the stress hormone cortisol in the forest walkers. Some reported reduced blood pressure and heart rate. The mechanisms are not fully understood and the research has methodological limitations, but the presence of biologically active compounds in forest air is not in dispute. Some terpenes have measurable effects on immune cells in laboratory conditions. Whether forest walks deliver enough of these compounds to affect human physiology significantly remains an open scientific question. The moss that carpets much of the forest floor is not a single kind of organism but a highly diverse group. There are around 12,000 species of mosses known to science, and they occupy almost every terrestrial environment on Earth, including the deep forest floor. Mosses are among the simplest of land plants. They lack the vascular system of xylem and phloem that allows trees and ferns to move water long distances. Because of this, they stay small, and they depend on absorbing water directly through their leaf surfaces. In the moist, dim environment of the forest floor, this is not a disadvantage. Water is plentiful and sunlight is scarce. Mosses can photosynthesize efficiently at very low light levels. Forest moss is extraordinarily dense with life. The individual moss plants form a 3-dimensional structure of tiny stems and leaves. Within this structure, an entire community of organisms lives. Tardigrades, sometimes called water bears, are 8-legged microscopic animals that live in the water films coating moss leaves. Tardigrades are among the most resilient animals known to science. When their environment dries out, they enter a state called cryptobiosis, in which all metabolic processes stop, and the animal can survive temperatures close to absolute zero and doses of radiation that would kill any other animal. When water returns, the tardigrade rehydrates and resumes its normal life. In the cyclically wet and dry environment of forest moss, tardigrades cycle in and out of cryptobiosis regularly. The forest floor is also the domain of ferns. Ferns are more complex than mosses. They have a vascular system and produce true roots, stems and leaves. But unlike the seed plants above them, ferns reproduce using spores rather than seeds. On the undersides of fern fronds, small structures called sori contain clusters of sporangia. These are tiny capsules filled with spores. When the spores are ripe, the sporangia open and the spores are released into the air. Each spore is tiny enough to travel in air currents for long distances. When a spore lands in a suitable moist location, it germinates into a tiny structure called a gametophyte. The gametophyte is a small flat green structure, usually no more than a centimetre across, that is quite different in appearance from the fern it will eventually produce. On the surface of the gametophyte, male and female reproductive cells are produced. When the gametophyte is wet, the male cells swim across the water film to fertilise the female cells. From the resulting fertilised cell, a new fern plant grows. This two-stage life cycle, alternating between a spore-producing stage and a gamete-producing stage, is the defining feature of the life cycle of all land plants. In mosses, the gametophyte stage is the dominant green plant that you see. In ferns, the sporophyte stage, the familiar fern frond, is the dominant stage. In seed plants, the gametophyte stage has been reduced to just a few cells inside the pollen grain and inside the ovule. The entire evolutionary history of land plant reproduction is a story of the gradual reduction of the gametophyte stage and the increasing independence of the sporophyte stage from the need for liquid water for fertilization. Insects are the most diverse group of animals in the forest. In a single hectare of old-growth forest, many thousands of insect species may coexist. Most of them are invisible most of the time. They live under bark, inside rotting logs in the soil, in leaf litter, inside plant tissue, or in the cups and crevices where water collects. The diversity of insect life in a forest reflects the diversity of the forest structure itself. Each layer of the forest, from deep soil to upper canopy, has its own characteristic insect community. Many insect species are specialists, dependent on a single host plant species or a single microhabitat. This specialization means that the loss of any tree species from a forest has cascading effects on the insect community. Insects that depend on that tree species either disappear or must shift to other hosts. Beetles are the most diverse order of insects, and the forest contains an enormous variety of beetle species. Ground beetles patrol the leaf litter hunting smaller invertebrates. Bark beetles tunnel beneath the bark of trees, sometimes transmitting fungal pathogens as they travel. Longhorn beetles lay eggs in the wood of dying or dead trees, and their larvae spend months or years tunneling through the wood, digesting it with the help of gut microbes. Dung beetles process the droppings of forest mammals. Leaf beetles eat the foliage of specific plant species. Fireflies, which are also beetles, produce their light through a chemical reaction in specialized cells in their abdomens. The light serves as a mating signal, and each species has its own specific pattern of flashes. Deadwood is one of the most important habitats in a deep forest. A fallen tree or a standing dead trunk, called a snag, provides habitat for hundreds of species that cannot survive in living forest without it. Fungi colonize dead wood and begin breaking it down. Bacteria follow. Beetles arrive to lay eggs. Woodpeckers excavate cavities in soft snags. Other birds and mammals use woodpecker cavities as nesting sites after the woodpeckers move on. Salamanders shelter in the cool, damp space beneath fallen logs. Millipedes and isopods consume the partially decayed wood. Mosses and lichens coat the outer surfaces. Over decades, a large dead tree passes through stages of decay, each stage dominated by a different community of organisms. The slowest stage of decay involves the breakdown of lignin, the tough polymer that gives wood its structural strength. Only certain fungi, called white-root fungi, have the enzymes needed to fully break down lignin. Lignin is one of the most chemically complex biological molecules. It is a polymer made of phenylprovenoid units linked in irregular and highly variable ways. Unlike cellulose, which has a regular repeating structure, lignin has no simple pattern. This chemical complexity is what makes it so resistant to decay. The white rot fungi that could break it down evolved this ability relatively recently in geological terms. Before white rot fungi evolved, dead wood accumulated without being broken down. Some researchers have proposed that this accumulation of undecomposed wood contributed to the formation of coal deposits during the Carboniferous period. When forests died and fell without organisms capable of breaking down lignin, organic matter piled up and was eventually buried and compressed into coal. The evolution of a lignin-digesting fungi may have brought this accumulation to an end. The deep forest at night is different from the deep forest during the day. Many of the animals that are invisible during daylight hours emerge after dark. Nocturnal animals are adapted to function in low light. Owls have eyes that are highly sensitive to light. The pupils of an owl's eyes can open extremely wide to allow in as much light as possible. The retina of an owl eye contains a high density of rod cells, which are the photoreceptors sensitive to dim light. Owls also have an asymmetrical arrangement of their ear openings, which allows them to locate sounds in 3 dimensions by detecting tiny differences in the time at which a sound reaches each ear. An owl can locate a mouse moving beneath snow in complete darkness using sound alone. Night mammals navigate using a combination of senses. Most small nocturnal mammals have well-developed whiskers called vibrissae. These are sensory organs, not simple hairs. Each vibrissa connects to a rich network of nerve endings at its base in the skin. The vibrissae detect air movements and physical contacts that give the animal information about its immediate environment in darkness. Many small forest mammals also produce and detect ultrasonic sounds. Shrews, which are insectivorous mammals common in forest undergrowth, produce high-frequency sounds and may use the echoes to navigate and locate prey in a manner similar to the echolocation of bats, though less sophisticated. Bats are among the most effective nocturnal hunters in the forest. Most forest bat species use echolocation to navigate and hunt. They produce short pulses of ultrasonic sound through their mouths or through specialized structures in their noses. These pulses travel through the air, reflect off objects and insects, and return to the bat's large mobile ears. The bat's brain processes the returning echoes to construct a detailed map of its surroundings. The time delay between the emitted pulse and the returning echo tells the bat how far away an object is. The frequency shift caused by the Doppler effect tells the bat whether the object is moving toward or away from it, and how fast. Different bat species have evolved different echolocation frequencies and pulse structures suited to their specific hunting environments. Forest bats, hunting among dense vegetation, use shorter pulses at higher frequencies than bats hunting in open air above the canopy. The sounds of the forest at night form a layer over the silence. Crickets and katydids sing. Some produce sound by rubbing the edge of one wing against a file-like structure on the other wing, a process called stridulation. The frequency of the cricket's song varies with temperature. As temperature rises, the chemical reactions driving the muscle contractions that produce the song speed up and the pulse rate increases. This relationship is consistent enough that some naturalists have used cricket chirp rates to estimate the outdoor temperature. Tree frogs call from branches near forest streams. Each species produces a call of a specific frequency and pattern that females of the same species recognize. Populations of the same frog species in different forest patches sometimes develop slightly different call patterns over generations, an acoustic divergence that can eventually contribute to speciation. Forest streams are corridors of a different kind of life within the deep forest. They bring oxygen-rich cold water from higher elevations. The water is typically clear because the forest floor filters the runoff. Stream banks are kept shaded and cool by the trees above. The result is a habitat for cold water species that cannot survive in warmer lowland waters. Salamanders lay their eggs under flat rocks in shallow streams. Caddisfly larvae construct protective cases from tiny grains of gravel or fragments of leaf, cemented together with silk they produce from glands near their mouths. These cases are carried by the larvae as they walk along the stream bottom, grazing on algae and organic matter. Stonefly nymphs require cold, well-oxygenated water and are often used as indicators of stream health. If stonefly nymphs are present in a stream, it generally means the water is clean and well oxygenated. The relationship between the forest and the stream runs in both directions. The forest shades the stream and keeps it cold. It provides fallen logs and branches that create complex habitat structure in the stream. Leaves fall into the stream and provide the primary food source for many stream invertebrates. The debris dams formed by fallen wood slow the current and create pools. These pools are important resting and feeding areas for fish and salamanders. In return, the stream influences the forest along its banks. Floods deposit nutrient-rich sediment on the floodplain. Trees that fall into the stream are stabilized by water and can persist for decades, slowly releasing nutrients as they decay. To animals that live in the stream carry nutrients from the water to the land. Dippers, small birds that walk along stream bottoms catching aquatic invertebrates, carry stream-derived nutrients onto the struss bank in their droppings. In forested regions where salmon migrate upstream to spawn, the connection between the forest and the stream is especially visible. Salmon spend years feeding in the ocean, accumulating marine-derived nutrients in their flesh. When they return to forest streams to spawn and die, their bodies deliver a pulse of marine nutrients to the forest ecosystem. Bears, wolves, eagles, and ravens carry salmon carcasses into the forest. The remains decay and enter the soil. Studies using the isotopic signature of marine nitrogen, which is distinct from terrestrial nitrogen, have detected marine-derived nitrogen in the wood of trees growing near salmon-bearing streams. The forest alongside these streams grows faster than the forest, farther from the salmon streams. The trees are taller, and their annual growth rings are wider in the years following large salmon runs. Amphibians are a particularly important, but often overlooked part of the deep forest community. Salamanders in particular reach high densities in many forest types. In the Appalachian forests of eastern North America, the total biomass of salamanders per hectare exceeds the total biomass of birds. Salamanders are predators of invertebrates, and as such they have significant effects on the invertebrate community. They also serve as prey for many other forest animals. Because they must return to water, or at least to moist environments to breed, they are sensitive indicators of forest condition. Their permeable skin makes them vulnerable to changes in soil chemistry and humidity. When a forest is logged or disturbed, salamander populations typically decline rapidly. When the forest recovers, the salamanders return, but recovery can take decades. Some species of forest salamanders have evolved to bypass the aquatic larval stage entirely. Lungless salamanders in the family Plethodontidae lay eggs under logs and stones in the forest. A parent coils around the eggs and stays with them until they hatch. The larvae complete their development inside the egg, emerging as fully formed miniature adults. This developmental strategy makes these salamanders entirely independent of streams or ponds. They can live in forests far from water. Because they breathe entirely through their moist skin and the lining of their mouths rather than through lungs, they require consistently moist environments. Their distribution within the forest is shaped by the humidity gradient, with highest densities in the dampest areas near streams and seeps. The deep forest is also a place of chemical signalling. Plants communicate with each other and with animals through chemical compounds released into the air. When a tree is attacked by insect herbivores, it produces volatile compounds called herbivore-induced plant volatiles. These compounds drift to neighbouring trees and can trigger defensive responses in those trees before the insects arrive. The mechanism is not electrical or nervous, it is chemical. The neighbouring tree detects the compounds through receptors in its leaf tissue and begins producing more kinds of chemicals in advance of the attack. This process has been documented in multiple forest tree species, including poplar, birch, and maple. The same volatile compounds that signal neighbouring trees also attract the natural enemies of the attacking insects. When caterpillars begin feeding on a plant, the plant releases specific blends of volatile compounds. These blends are detected by parasitic wasps that prey on caterpillars. The wasps follow the chemical trail to the caterpillar-infested plant and lay their eggs inside the caterpillars. The wasp larvae then develop inside the caterpillar and eventually kill it. The plant is, in a sense, calling for help by releasing a signal that attracts the enemies of its attackers. This three-way interaction between plant, herbivore, and natural enemy of the herbivore has been studied extensively, and the precision of the chemical signals is remarkable. Different attacking species can trigger the production of different signal blends. Plants also signal through root chemicals. When a plant is attacked by soil pathogens, it can release compounds through its roots that attract specific beneficial bacteria. These bacteria produce antibiotics and other compounds that suppress the pathogen. The plant is actively recruiting a microbial defense force by releasing chemical attractants through its roots. The composition of the microbial community in the rhizosphere changes measurably in response to plant stress. This suggests that the relationship between plants and their root-associated microbes is not static, but dynamic, changing in response to threats. The air above a deep forest is also full of biological particles. Pollen from flowering trees travels in the air during the brief periods when each tree species flowers. Each species has a characteristic pollen grain shape and size. The pollen grains of oak are small and smooth for efficient wind dispersal. Those of hazel are smooth and rounded. Those of pine have two air bladders that increase their buoyancy in air. By collecting and examining these pollen grains from sediment layers in lake bottoms, scientists can reconstruct the history of the surrounding forest over thousands of years. Pollen accumulates in lake sediment year by year. By dating the sediment layers and identifying the pollen types present in each layer, researchers can determine which tree species were present in the region at different times in the past. Spores of mosses, ferns, and fungi also travel through forest air. Fungal spores are produced in enormous quantities. A single puffball mushroom can contain several trillion spores. The timing of spore release in many fungal species is timed to coincide with the most favourable conditions for dispersal. Some fungi release their spores preferentially when the air is still and humidity is high, conditions that favour spore survival in the air. Others release spores in response to dew or rain. The splash of a raindrop on a mature mushroom cap can scatter spores in all directions. Some fungi have mechanisms to actively shoot spores into the air. Ascomycete fungi, which include morel mushrooms and cup fungi, build up internal pressure inside their spore-bearing cells and explosively discharge spores at high velocity when conditions are right. Birds are among the most visible residents of the deep forest, but different bird species occupy different layers of the forest structure. Warblers forage in the upper canopy. Thrushes move through the understory. Ovenbirds walk on the forest floor searching through the leaf litter. Each species is adapted to its preferred layer. Their bill shapes, leg lengths, and foraging behaviours reflect their particular niche. In a mature forest with all layers intact, the bird community is correspondingly diverse. When any layer is removed, the species associated with that layer disappear. In a forest that has lost its large old trees, the species that depend on tree cavities disappear. When the understory shrubs are removed, the ground nesting birds that require low shrub cover for nesting abandon the area. Migration brings a seasonal pulse of energy through the forest. In spring, warblers, flycatchers, thrushes and vireos arrive from tropical wintering grounds to breed in temperate forests. They have travelled thousands of kilometres by night, navigating by the stars and by the magnetic field of the Earth. The arrival of migrants roughly coincides with the emergence of the insects on which they feed. This timing is not random. It has been shaped over thousands of generations of selection for birds that arrive when food is most available. In recent decades, some studies have shown that the timing of insect emergence has shifted earlier in some regions due to warming temperatures. If the timing of migrant bird arrival does not shift correspondingly, arriving birds may find that the peak of insect abundance has already passed, reducing their breeding success. The forest at its largest scale is a system that influences weather. Large forests generate what some atmospheric scientists call biotic pump effects. The continuous transpiration of a large forest releases water vapour into the atmosphere. This water vapour condenses to form clouds and eventually falls as rain. The rain is then absorbed by the forest soils, taken up by roots, and transpired again. In tropical regions, this cycle can sustain rainfall deep into continental interiors. The Amazon rainforest generates what are sometimes called flying rivers, enormous quantities of water vapour that travel westward through the atmosphere, eventually precipitating as rain over the Andes and contributing to rivers that flow back into the Atlantic. The water cycles around and around, moved by the forest itself. Some climatologists have proposed that large intact forests are not merely passive recipients of rainfall, but active generators of the moisture that maintains them. Soil bacteria in the deep forest perform chemical transformations that are invisible but essential to how the forest functions. Nitrifying bacteria convert ammonia into nitrite and then into nitrate, the form of nitrogen most easily absorbed by plant roots. Denitrifying bacteria perform the reverse process, converting nitrate back into atmospheric nitrogen gas under low oxygen conditions. This denitrification can be important in waterlogged forest soils where oxygen is scarce. The balance between nitrification and denitrification in forest soils determines how much nitrogen is retained in the ecosystem versus how much is lost to the atmosphere. Nitrogen is often the limiting nutrient in temperate forest growth. Forests with high rates of denitrification may lose nitrogen faster than it's replaced and grow more slowly as a result. Archaea, which are single-celled microorganisms distinct from both bacteria and eukaryotes, are present in forest soils in large numbers, but are less well studied than bacteria. Some archaea in forest soils participate in nitrogen cycling. A group called ammonia-oxidizing archaea can oxidize ammonia to nitrite, contributing to the nitrification process. In some forest soils, archaea may carry out more nitrification than bacteria. The relative contributions of bacteria and archaea to soil nitrogen cycling appear to vary with soil chemistry, temperature, and moisture. Cold and acidic forest soils, such as those under spruce forests at high elevation, tend to be more dominated by archaea in their nitrification activity. Viruses are the most numerous biological entities in forest soil. In 1 milliliter of forest soil, there can be tens of millions of virus particles. Most of these viruses infect bacteria and archaeopodra rather than animals or plants. They are called bacteriophages, or simply phages. Phages play a significant role in controlling bacterial populations. When a bacterial population grows large, phage populations also grow, infecting and killing bacteria. This predation prevents any single bacterial species from dominating the soil community entirely. Phages also transfer genetic material between bacteria, a process called transduction. A phage that has infected one bacterium and incorporated some of its genetic material can carry that material to a new bacterial host. This transfer of genes between bacteria via phages is one of the ways that metabolic capabilities spread through soil bacteria communities. The soil fauna, meaning the animals that live in soil, form a complex food web. Bacteria and fungi are consumed by protozoa and nematodes. These in turn are consumed by larger nematodes, by mites, and by small beetles. Centipedes hunt in the larger pores and channels of the soil. Ants and earthworms restructure the physical architecture of the soil as they dig. The mixing of soil by animals is called bioturbation. Earthworms pull organic matter from the surface into deeper soil layers as they feed. Their burrows improve soil drainage and aeration. The passage of soil through an earthworm's gut accelerates the decomposition of organic matter and increases the availability of nutrients. Charles Darwin spent the last years of his life studying earthworms and their effects on the English countryside. He estimated that the earthworms, a typical English meadow, passed all of the soil in the top layer through their bodies over the course of several decades. Ants are significant agents of soil modification in many forests. Wood ants build large mounds of plant material above nests that extend deeply into the soil below. The mound traps solar heat and keeps the nest warmer than the surrounding soil, extending the effective length of the growing season for the colony. The soil of an ant nest has a different chemical composition from the surrounding soil due to the organic matter brought into it and the concentrated biological activity within it. Trees sometimes grow more vigorously directly adjacent to ant mounds, apparently benefiting from the altered soil chemistry. The foraging activities of ants distribute seeds, carry insect prey across large areas, and contribute to the mixing of soil and organic matter throughout the forest floor. The water that collects in the crevices and cavities of forest trees supports communities of aquatic organisms far from any stream or pond. These tree holes, called phytotelmata, fill with rainwater and decaying leaf fragments. In this tiny enclosed world, bacteria decompose the organic matter. Mosquito larvae feed on the bacteria. Predatory insect larvae eat the mosquito larvae. Some species of tree hole organisms are found nowhere else. They've adapted entirely to the ephemeral and enclosed world of the tree cavity pool. The water chemistry of tree holes varies enormously from tree to tree and from species to species, depending on the chemistry of the bark and the rate of leaf decomposition. Some wood species produce water that's highly acidic. Others produce water nearly neutral in pH. The organisms adapted to each tree hole type are different accordingly. Lichens, which are found throughout the deep forest on bark, rocks and soil, are not single organisms but partnerships between fungi and photosynthetic partners. Most lichens contain a fungal partner and either a green alga, a cyanobacterium or both. The fungal part provides structure and protection. The photosynthetic partner provides sugars. The relationship was long described as mutualistic, with both partners benefiting. More recent research has suggested the relationship may be more complex. The fungal partner controls the growth and reproduction of the photosynthetic partner in ways that suggest a degree of exploitation. And some lichens appear to contain additional microbial partners, including basidiomycete yeasts that have only recently been discovered living within the lichen tissue. The internal ecology of a single lichen thallus, which is the flat or leafy body of the lichen, may be more complex than previously understood. Lichens grow extraordinarily slowly. A lichen crust on a forest rock may be decades or centuries old despite being only a few centimetres across. Growth rates of less than a millimetre per year are common in many species. Because of this, slow growth lichens can persist in stable environments for very long periods and accumulate information about long-term environmental conditions in their tissue chemistry. The age of a lichen can sometimes be estimated from its size using species-specific growth rate data, a technique called lichenometry that has been used to date exposed rock surfaces in alpine and arctic environments. The chemical complexity of forest organisms extends to their defensive compounds. Plants produce a vast array of secondary metabolites, chemicals that are not directly required for basic cellular functions, but serve defensive or other ecological roles. Alkaloids, terpenes, phenolics, and glucosinolates are among the major classes of plant defensive compounds. Each has evolved in response to specific threats. Some alkaloids are toxic to insects. Some terpenes repel or poison fungal pathogens. Some phenolics inhibit the germination of competitor plant seeds in the soil near the producing plant. The diversity of plant defensive chemistry in a mature forest is immense. A single tree species may produce dozens of different defensive compounds in different tissues at different times of year. Some defensive compounds in forest plants have effects on mammals, including humans. Aspirin is derived from salicylic acid, a compound produced by willow trees and other plants as a wound response signal. Many plant alkaloids are pharmacologically active in the human nervous system. Quinine, morphine, caffeine, and atropine are all alkaloids. derived from plants that evolved them for non-human purposes. The study of plant medicinal compounds, called ethnobotany and phytopharmacology, has drawn on the knowledge of indigenous cultures who identified plant species with useful properties over many generations of empirical observation. A significant proportion of the pharmaceutical compounds in use today are either directly derived from plant compounds or are synthetic analogues of plant compounds. The old trees of a deep forest are structures of great biological complexity. A very old tree is not simply a larger version of a young tree. Over centuries of growth, it develops features that young trees lack. Its bark becomes deeply furrowed, creating microhabitats for insect and lichens. Its crown becomes more irregular, with dead branches alongside living ones, creating structural diversity. Large cavities develop in the trunk and major branches, formed initially by branch loss and then enlarged by the action of fungi and woodpeckers. These cavities are essential nesting sites for owls, ducks, bats, and many other species. The tree's root system becomes intricately connected to the mycorrhizal network of the forest. Its soil footprint is large. Its contribution to soil organic matter is enormous. When an old tree finally dies, it does not disappear from the forest community. It simply transitions to a new role. As a standing dead tree, a snag, it provides nesting cavities for woodpeckers and other cavity-nesting birds. When it falls, it becomes a nurse log for mosses, ferns, and tree seedlings. In many forest types, the seedlings of the dominant tree species establish most successfully on the elevated, decaying wood of nursed logs, rather than on the flat forest floor. A nurse log provides drainage from standing water, reduced competition from ground plants, and a substrate free of some of the pathogens present in mineral soil. In a mature forest, the next generation of large trees often grows in a row that traces the line of a long-decayed nurse log. Even after the log has entirely decomposed, its influence persists in the arrangement of the living trees that replaced it. The forest is always changing. Even in the deepest, most undisturbed forest, things are in flux. Trees grow, some deramage, others are knocked down by windstorms. A fallen tree creates a gap in the canopy. Light streams through the gap and reaches the forest floor. Seeds that have been waiting in the soil seed bank germinate in response to the increased light. Seedlings grow quickly in the gap. Competition among the seedlings is intense. Within a few decades, one or two may have grown tall enough to begin filling the gap. Within a century, the gap may be closed again. These gap phase dynamics, the cycle of gap creation and closure, create a mosaic of patches of different ages and structures across the forest landscape. This mosaic of patch ages means that at any given moment, a large forest contains areas of young growth, middle-aged growth, and old growth, all intermixed. Each patch type supports a somewhat different community of organisms. Species that need dense shade and deep litter depend on the old growth patches. Species that need higher light levels and more open structure use the younger patches. The overall diversity of the forest at the landscape level is higher because of the structural diversity than it would be if the entire forest were of a uniform age. The soil seed bank is a hidden reservoir of forest history. Seeds from many species can persist in the soil for years or even decades before germinating. Some seeds have hard seed coats that must be physically abraded or chemically treated before they can absorb water and germinate. Others require a period of cold followed by warmth before germination can occur, a process called stratification. Still others require fire to germinate. their seed coats softened by the heat of a passing flame. This diversity of germination requirements means that the forest is perpetually ready to respond to a variety of disturbance types. After a fire-adapted species germinate quickly. After a windstorm that creates gaps, light-demanding species emerge from the seed bank. The chemical history of the forest is also recorded in its leaves. The isotopic composition of carbon in leaf tissue reflects the carbon dioxide available when the leaf was growing. The isotopic composition of nitrogen reflects the nitrogen sources available to the tree. The concentration of various trace elements in leaves reflects the chemistry of the soil in which the tree grew. By analysing these chemical signatures, researchers can reconstruct aspects of the growing environment with surprising precision. Ancient leaves preserved in lake sediments or in permafrost carry chemical signatures that allow scientists to infer past atmospheric carbon dioxide concentrations and past soil conditions. The diversity of deep forest life extends to forms of life that challenge the conventional categories of living organisms. Slime molds, for example, are organisms that spend part of their life cycle as individual amoeba-like cells and part as a coordinated multicellular body. When food is scarce, the individual cells aggregate and form a slug-like structure that moves toward light and eventually forms a fruiting body that releases spores. In some species, the aggregating cells produce a complex network structure as they move toward food sources that optimizes for efficient flow through the network. This self-organizing behavior in organisms with no nervous system has attracted interest from mathematicians and network scientists. The network architectures produced by slime molds optimizing their own nutrient transport have been compared to optimal designs for transportation networks in human cities. The variety of life strategies in the deep forest reflects the complexity of the environment and the length of time over which life in forests has evolved. The first land plants appeared roughly 470 million years ago. The first forests appeared perhaps 400 million years ago. In that time, organisms have evolved to exploit every available niche in the forest environment. The diversity we observe in a forest today is the product of hundreds of millions of years of evolutionary experimentation. Each organism present is the survivor of an unbroken chain of ancestors extending back to the earliest colonizers of the land. Each is finely tuned to its specific place in the forest community. The deep forest asks nothing of you. It existed before humans arrived, and it continues to exist in the places where it has been left undisturbed. Its processes proceed according to their own logic. The fungi extend their threads through the dark soil. The roots absorb and release. The leaves capture light and turn it into sugar. The fallen wood decays back into the soil that will nourish the next generation of trees. The birds move through the layers. The insects complete their cycles. The water moves from rain to root to leaf to air and back to rain again. In the forest, everything that falls is eventually lifted again. Everything that dies becomes part of something living. The carbon in a leaf that fell last autumn is already moving through the body of a fungus that is already feeding a root that is already drawing it upward toward the light. The atoms cycle. The patterns persist. The forest continues. Before we reach that rest, there is still much quiet science to explore. The forest offers more layers of slow truth than any single evening can exhaust. So let us continue walking through it, gently and without hurry. The photosynthetic pigments in forest leaves are not only chlorophyll. Alongside the green chlorophylls there are yellow and orange pigments called carotenoids. These include beta-carotene and xanthophylls. During the growing season, the carotenoids are masked by the abundance of chlorophyll. When autumn arrives and days grow shorter, the tree begins to withdraw nitrogen from its leaves back into the stem for storage. Chlorophyll breaks down faster than carotenoids when the supply of nitrogen is reduced. As chlorophyll disappears, the yellows and oranges of the carotenoids become visible. The red colours of autumn leaves are different in origin. They are produced by a class of pigments called anthocyanins, which are synthesized specifically in autumn. The production of anthocyanins is triggered by bright light and by cold temperatures acting on sugar-rich leaf tissue. On sunny autumn days with cold nights, the anthocyanins production is highest and the reds are deepest. The precise function of autumn red coloration is still discussed among biologists. Some research suggests it may signal to insects that a tree is well defended, discouraging insects from laying their eggs on that tree in autumn. Other research proposes it serves a photoprotective function, shielding the leaf tissue during the nutrient withdrawal process. The breakdown and reabsorption of chlorophyll in autumn represents a significant nutrient recovery operation. Nitrogen is often the element that most limits growth in temperate forests. Chlorophyll contains nitrogen. Before the leaf is dropped, the tree carefully disassembles the chlorophyll molecules and moves the nitrogen back into the woody tissue for storage over winter. This nitrogen will be remobilized in spring to build a next generation of leaves. A mature tree invests a great deal in recycling nitrogen from its leaves. Trees that do this efficiently can produce leaves at lower cost each spring. The efficiency of this recycling varies among species. Deciduous trees generally recover a higher fraction of leaf nitrogen before abscission than conifers, which retain their needles for several years and must invest differently in their nitrogen economy. The abscission of leaves in autumn is not a passive process, it is an active cellular event. At the base of each leaf stalk, called the petiole, there is a specialized layer of cells called the abscission zone. Before a leaf is dropped, the cells in this zone produce enzymes that break down cell walls between the abscission zone cells and the vascular connections between the leaf and the twig. As these connections weaken, the leaf becomes less firmly attached. The break-in of the leaf from the stem leaves a leaf scar on the twig. This scar is sealed by a layer of protective cell that prevent water loss and pathogen entry. The pattern of leaf scars on a twig is diagnostic for the tree species. The arrangement, shape and number of vascular bundle scars visible within each leaf scar are features used in botanical identification of deciduous trees in winter when no leaves are present. The winter dormancy of deciduous trees is a state of suspended but not absent activity. The tree is not dead during winter. Its roots continue absorbing water when the soil is not frozen. Mycorrhizal fungi remain active in the soil at temperatures just above freezing. The cambium is dormant but ready to resume division in spring. The buds that will become next year's leaves and flowers are already formed, having been produced the previous summer. They are wrapped in protective bud scales, which are modified leaves hardened and waxed against cold and desiccation. Inside the bud, the embryonic leaf tissue is compressed. Each bud contains the complete blueprint for a shoot that will emerge in spring, develop, extend and produce all the leaves and branches it was programmed to produce. The timing of bud burst in spring is controlled by a combination of temperature and day length. Most temperate tree species require a period of cold, called chilling, before they can respond to warming temperatures in spring. This chilling requirement prevents the tree from breaking dormancy during a brief warm spell in mid-winter. Only after the chilling requirement has been met does the tree begin monitoring warming temperatures. When accumulated warmth above a threshold temperature reaches a certain total, the buds burst. The precision of this timing is remarkable. Different tree species break dormancy at different times, and the sequence is consistent from year to year in the same forest. Oak is typically one of the last to leaf out. Birch and willow are among the first. In between are dozens of other species, each following its own temperature schedule. Conifers, which dominate many of the world's deep forests, particularly at higher latitudes and elevations, have a quite different relationship with seasonality. Most conifers retain their needles through the winter. Their needles are adapted to minimize water loss in cold conditions. The stomata on conifer needles are sunken into grooves and can close tightly to prevent desiccation when the ground is frozen and water cannot be absorbed. The waxy coating on needle surfaces reduces water loss further. Conifer needles can tolerate temperatures far below zero without cell damage, because the water in the cells contains dissolved solutes that depress the freezing point, and because the cells can also supercool, that is, remain liquid below the normal freezing point by excluding the ice crystal nuclei that would trigger freezing. Larches are a notable exception among conifers. They are deciduous conifers, shedding their needles each autumn and regrowing them each spring. Larches grow in some of the coldest forested regions on Earth, including the boreal forests of Siberia, where temperatures can fall below -50 degrees Celsius. The deciduous habit of larches may be an adaptation to these extreme cold conditions. By shedding their needles, the trees avoid the damage that might occur to photosynthetic tissue during the deepest cold, and also reduce the snow load on branches, lowering the risk of breakage under heavy snowfall. The boreal forest, called the taiga, is the world's largest terrestrial biome. It extends in a broad belt across northern North America, Europe, and Asia. The trees of the taiga are few in species but vast in number. Black spruce, white spruce, jack pine, lodgepole pine, Scots pine, Siberian larch, and a handful of other species cover millions of square kilometers. The simplicity of tree species composition does not mean the taiga is a simple ecosystem. The understory of mosses, lichens, and low shrubs is diverse. The soil communities are complex. The animal communities are adapted to extreme seasonality and cold. Black spruce of the North American taiga grows in particularly challenging conditions. It can grow on water-saturated soils where few other trees can survive. Its shallow root system spreads horizontally just below the surface, above the permafrost that lies beneath. In winter, the water in the upper soil freezes solid. The roots are embedded in this frozen layer and cannot take up water. The needles, though evergreen, cannot photosynthesize efficiently in extreme cold. The tree essentially waits. In the brief warm season of high northern latitudes, the soil thaws just enough for the roots to absorb water. The tree photosynthesizes rapidly during the long days of the subarctic summer and stores the products. Then cold returns and it waits again. Permafrost, the permanently frozen soil of Arctic and subarctic regions, contains this enormous quantities of organic carbon. The cold, waterlogged conditions of permafrost soils slow decomposition so severely that organic material can accumulate for thousands of years without fully breaking down. Some permafrost soils contain organic carbon that was deposited during the last ice age. preserved for more than 10,000 years by continuous freezing. The total amount of carbon stored in permafrost soils globally is estimated to be roughly 1.5 trillion metric tons, which is roughly twice the amount of carbon currently in the atmosphere as carbon dioxide. As temperatures rise in the Arctic, some of this permafrost is thawing. When it thaws, the previously preserved organic matter becomes available to microbial decomposition. This decomposition releases carbon dioxide and methane, both greenhouse gases. This could create a feedback in which warming causes permafrost thaw, which causes more warming. The trees of the deep forest interact with the atmosphere not only through photosynthesis and transpiration, but also through other chemical exchanges. Some trees release isoprene, a highly reactive 5-carbon molecule, through their leaves. Isoprene production increases with temperature and light. In warm, sunny conditions, a large deciduous forest releases enormous quantities of isoprene into the atmosphere. In the atmosphere, isoprene reacts rapidly with hydroxyl radicals, which are key oxidizing agents in atmospheric chemistry. This reaction depletes the concentration of hydroxyl radicals in the atmosphere above forests. Because hydroxyl radicals are responsible for breaking down methane, which is a potent greenhouse gas, the depletion of hydroxyl radicals by forest isoprene emissions could potentially allow methane to accumulate in the lower atmosphere. The net effect of forest isoprene emissions on climate is complex and not fully resolved. The forest influences the colour of the sky. As noted earlier, the terpene compounds released by forests scatter light and can give a bluish-hazed forested mountains. But forests also influence atmospheric aerosol chemistry more broadly. The oxidation products of forest-emitted terpenes can condense to form small particles called secondary organic aerosols. These particles serve as nuclei for water droplet condensation and can influence cloud formation. Clouds over forests may form at lower water vapor concentrations than clouds over areas without forests, because the aerosol particles provide more condensation nuclei. This means forests may enhance cloud formation and precipitation in ways that go beyond simple water vapor contributions. The effect of forests on regional climate through changes in surface properties is also significant. The albedo of a surface, which is the fraction of incoming sunlight it reflects, is different for forests than for grasslands, croplands, or bare soil. A dark conifer forest has a very low albedo. It absorbs more sunlight than it reflects. In summer, this absorption warms the surface and the air above it. In regions where conifers replace snow-covered ground in spring, the dark trees absorb more heat than the snow would have reflected, warming the local climate. In tropical regions where forests have high albedo and also release large quantities of water vapor through transpiration, the net effect on climate is different. The complex interactions between surface albedo, water vapor, aerosols, and cloud formation mean that the climate effects of forests vary by region and by season. The study of how forests interact with the atmosphere and climate is part of a broader field called Earth System Science. In this framework, the forest is not merely a collection of biological organisms, but a component of the planetary system that processes energy and matter. The carbon fixed by photosynthesis in forests contributes to the regulation of atmospheric carbon dioxide. The water cycled through forests contributes to the regulation of the hydrological cycle. The trace gas emissions of forests affect atmospheric chemistry. The surface properties of forests affect the energy balance of the land surface. Everything connects to everything else in a system of feedback and regulation that have been operating for hundreds of millions of years. The relationship between deep forest life and geological time is profound. The forest soils building today are the product of millions of years of biological activity. The coal seams mined for energy represent forests that died hundreds of millions of years ago. The calcium in forest soils comes in part from the slow weathering of rocks by the acids produced by tree roots and soil organisms, a process called biological weathering that has been operating since plants first colonized the land, and that has slowly changed the chemistry of the oceans and atmosphere over geological time. The forests are not a feature added onto the planet. They are a deep part of the planet's chemistry and history. Rock weathering by plant roots and soil acids is one of the major mechanisms by which carbon dioxide is removed from the atmosphere over geological timescales. When a root or a soil acid reacts with a silicate mineral in rock, the chemical reaction consumes carbon dioxide and produces bicarbonate ions that eventually wash into rivers and oceans. In the ocean, this bicarbonate is incorporated into the shells of marine organisms and eventually into limestone rock on the ocean floor. Over millions of years, this process has drawn down large amounts of atmospheric carbon dioxide. The biological colonization of the land by plants roughly 470 million years ago may have substantially accelerated rock weathering and contributed to a long-term decline in atmospheric carbon dioxide that cooled the planet. The diversity of tree root architectures and different species reflects different strategies for acquiring resources from the soil. Some trees produce deep taproots that penetrate far below the surface, accessing water from deep soil layers that are not affected by surface drought. Others produce wide-spreading, shallow root systems that quickly intercept nutrients and water from the upper soil layers, where organic matter is most concentrated. The pine has a root system that combines a taproot with many lateral roots. The shallow-rooted spruce spreads its roots widely, but not deeply. The beech develops a deep, wide-spreading root system that can extend well beyond the crown of the tree. The fine roots at the tips of these systems are ephemeral, growing rapidly in response to pockets of available nutrients and dying back when the nutrients are depleted. A single large tree may produce hundreds of kilometers of fine roots in a single growing season, most of which will die within weeks or months. The turnover of fine roots is a significant pathway of carbon into the soil. When fine roots die, they decompose and their carbon enters the soil organic matter pool. In many forests, the annual input of carbon from root turnover equals or exceeds the annual input of carbon from leaf litter. This underground contribution to soil carbon is harder to measure than the leaf litter input because it cannot simply be collected in a trap. Researchers estimate fine root production by periodically sampling small cores of soil and counting the living and dead roots present, or by installing transparent tubes called rhizotrons into the soil and photographing the roots growing against the tube wall over time. The interaction between tree roots and soil animals is also significant. Earthworms are attracted to the vicinity of tree roots because roots create channels in the soil, and because root exudates provide organic compounds. Earthworm activity in turn benefits tree growth by improving soil structure and nutrient availability. This mutual enhancement creates a positive feedback. More earthworms improve soil conditions for roots. Better root growth provides more resources for earthworms. In forests where earthworms are absent, such as in regions where they were excluded by glaciation during the last ice age, their introduction can dramatically alter the forest floor ecosystem. In northern North American forests that were glaciated, non-native European earthworms introduced accidentally by humans have disrupted the native forest floor communities by consuming the thick layer of organic matter on which native forest floor plants depended. The way that animals move through the forest at night is shaped as much by smell as by any other sense. Mammals communicate through chemical signals called pheromones and through general olfactory cues. The forest is saturated with chemical information that is invisible to most human observers, but deeply meaningful to the animals that live there. A deer moving through the forest at night follows scent trails left by other deer, detects the scent of predators, locates food plants by smell, and leaves its own scent on vegetation through glands in its hooves and on its face. The scent environment of the forest changes with weather. Rain washes scent from surfaces. Cold air suppresses the evaporation of volatile compounds. On warm, still nights, scent trails persist longer, and the olfactory landscape becomes richer. Foxes hunting at night in the forest use smell to locate earthworms near the soil surface on wet nights. They move slowly and stop frequently, lowering their heads to sniff the ground. Their hearing is also acute. The rustling of a beetle in the litter at several meters distance is detectable. The direction of a sound source is calculated from the tiny time difference in arrival between the two ears, a computation that is performed rapidly and automatically by auditory circuits in the brain. Foxes and many other predators can also detect the low-frequency seismic vibrations produced by animals moving underground, picking up vibrations through bones in the skull connected to the middle ear. The plant communities of the deep forest floor are a specialized group. Few plant species can photosynthesize effectively at the very low light levels present beneath a closed forest canopy. Those that do have evolved specific adaptations. Shade-tolerant plants typically have larger and thinner leaves that maximize light capture per unit of leaf material. They often position their leaves horizontally to intercept as much of the diffuse light as possible. Some shade plants have leaves arranged in a flat mosaic pattern in which the leaves of a single plant tile the ground without overlapping, minimizing self-shading. Some forest floor plants have completely abandoned photosynthesis. Ghost pipes, also called Indian pipes or Monotropa uniflora, are white plants that lack chlorophyll entirely. They cannot photosynthesize, as instead they obtain all their carbon from the mycorrhizal fungal network, which in turn obtains it from the photosynthesizing trees connected to that network. Ghost pipes are therefore parasitic on the mycorrhizal network. They are sometimes described as mycoheterotrophs. They represent an evolutionary transition away from autotrophy toward full dependence on the fungal carbon pipeline. Other mycoheterotrophic plants in forest floors include some orchid species that begin their lives entirely underground without chlorophyll and only develop green tissue once they emerge above ground, if they emerge at all. Some underground orchid species spend their entire lives below ground, flowering and setting seed without ever producing green tissue. The orchid family is extraordinarily diverse, and many of its members have highly specific mycorrhizal associations. Unlike most plants, orchids cannot germinate and establish without their specific fungal partner. The tiny dust-like seeds of orchids contain almost no stored food. When a seed germinates, it immediately requires fungal hyphaeas to penetrate its cells and provide sugars before the seedling can develop. This absolute dependence on fungi from the first moment of life means that orchids can only grow where their specific fungal partners are present. This is one reason why orchids are so difficult to transplant. Moving an orchid to a new location means also ensuring that the specific fungal partner is present in the new soil. Many horticultural attempts to establish wild orchids in new locations fail because the fungal partner is absent. The flora of a tropical deep forest is different from a temperate deep forest in fundamental ways. In the tropics, there is no winter dormancy. Photosynthesis proceeds year-round. Decomposition is rapid because the warm, moist conditions favour microbial activity. The rate of nutrient cycling is fast. Leaf litter on the floor of a tropical forest decomposes within months. The nutrients released move quickly into living tissue rather than accumulating in thick organic layers as in boreal forests. The soil of many tropical forests contains relatively little stored organic matter compared with temperate or boreal forests. The nutrients of the tropical forest are concentrated in the living biomass, the trees, and their associated organisms rather than in the soil. When tropical forest is cleared, the nutrients that were in the living trees are removed or burned, and the soil beneath may be relatively nutrient poor, unsuitable for sustained agriculture without heavy fertilization. The canopy of a tropical forest harbours a particularly extraordinary diversity of life. Tropical forest canopies contain thousands of species of insects, birds, frogs, lizards, and mammals that never or rarely descend to the ground. Some of these canopy animals are entirely unknown except from specimens collected in the forest canopy using fog machines that knock insects from the treetops, or from observations made from canopy platforms and walkways. Arthur Tansley, who coined the term ecosystem in 1935, recognized that a forest is not simply a collection of plants but a complete system of interacting organisms and their physical environment. The canopy of a tropical forest demonstrates this perhaps more vividly than any other environment on Earth. It's a three-dimensional habitat of staggering complexity. The sounds of a tropical forest at night are different from those of a temperate forest. The density of calling insects, frogs and birds creates a continuous acoustic layer that can be described as the soundscape or acoustic environment of the forest. Ecoacoustics, the study of soundscapes in natural environments, has developed methods for quantifying the acoustic diversity and complexity of forest soundscapes. Some research suggests that the complexity and richness of a forest soundscape correlates with the biodiversity of the ecosystem. A degraded forest with fewer species produces a simpler, less rich soundscape. Conservation biologists have begun using passive acoustic monitoring, networks of microphones left recording automatically to assess the health of forest ecosystems and detect changes over time. The ancient forest that once covered large parts of the earth looked quite different from modern forests. The first forests, which appeared roughly 400 million years ago in the Devonian period, contained tree-like plants quite different from any living today. Archaeopteris was a large tree with a woody trunk and fern-like fronds. Lepidodendron, which dominated the coal swamp forests of the Carboniferous period, was a tree-like lycotsade related to modern club mosses. It grew to heights of 30 metres or more and had a distinctive diamond-patterned bark made of the scars of fallen leaves. These ancient forest trees reproduced by spores rather than seeds and had life strategies quite different from modern trees. The understanding of these extinct forests comes from the fossil record preserved in coal measures and ancient soils around the world. The transition from spore-bearing to seed-bearing plants, which occurred in the late Devonian and Carboniferous periods, was one of the major transitions in the history of life. Seeds allowed plants to reproduce without liquid water for fertilization. The embryo inside a seed is protected by a tough coat and supplied with stored food to support the first stages of growth. Seeds can be dispersed by wind, water, and animals to new locations where they can remain dormant until conditions favor germination. The seed was an evolutionary innovation that allowed plants to colonize drier and more unpredictable environments, and that ultimately led to the dominance of seed plants in almost all terrestrial ecosystems on Earth today. The diversification of flowering plants, the angiosperms, which began in the Cretaceous period around 140 million years ago, transformed the world's forests. Before the angiosperms, forests were dominated by gymnosperms, including conifers, cycads, and their relatives. Gymnosperms produce naked seeds in cones or on modified leaves. Angiosperms enclose their seeds in fruits and produce flowers to attract animal pollinators. The evolution of flowers and the coevolution of insect pollinators was one of the great biological interactions of Earth history. Flowers evolved structures that attracted specific insect species. Insects evolved sensory systems and body structures suited to visiting specific flower types. This mutual specialization drove both plant and insect diversification. The extraordinary diversity of both flowering plants and insects today reflects in part the long history of this co-evolutionary relationship. Pollination in forest trees takes various forms. Wind-pollinated trees such as oaks, beeches, and conifers produce enormous quantities of pollen because most pollen grains never reach a receptive flower. The probability of any individual pollen grain landing on the right flower is very low, so the tree compensates by producing vast numbers. A single oak tree can produce several million pollen grains per day during the pollination season. Animal-pollinated trees produce less pollen, but invest more in attracting pollinators through flowers with colours, scents, and nectar. The investment in pollen transport is outsourced to the animal. This is generally more efficient than wind pollination when pollinators are reliable and when trees of the same species are not too scarce. Seed dispersal in deep forest trees is as varied as pollination. Many forest trees produce fruits eaten by animals. The animal digests the fruit flesh and deposits the seed, often far from the parent tree in its droppings. Jays bury acorns and beech nuts as winter food stores and forget or abandon many of these caches. The forgotten caches germinate and produce new trees. In this way, jays actively expand the distribution of oak and beech trees across the landscape. The co-evolutionary relationship between jays and oaks is an example of a mutualism in which both species benefit. The jay obtains food and the oak obtains dispersal. However, the relationship is not perfectly mutualistic. The jay benefits most when it retrieves its caches. The oak benefits most when the caches are abandoned. There is a degree of conflict embedded within the cooperation. The nuts and fruits produced by forest trees are among the richest sources of stored energy in the forest ecosystem. They represent a concentration of carbohydrates, fats, and proteins that took months of photosynthetic work to accumulate. Many forest animals time their most intense foraging activity to the autumn mast, the period when trees produce their annual crop of nuts and seeds. Bears fatten rapidly on beech nuts and acorns in autumn. Deer shift their diets toward mast crops. Squirrels and chipmunks harvest and store enormous quantities. Wood mice cache seeds underground. Even some forest floor invertebrates consume large numbers of fallen seeds. The energy that moves through the forest food web in autumn during the mast season is disproportionately large compared with the energy available at other times of year. Masting, the synchronized production of large seed crops by forest trees, is a fascinating ecological phenomenon. In mast years, oak trees or beech trees across entire regions produce vast quantities of nuts simultaneously. In non-mast years, the same trees produce few or no nuts. This synchronization occurs over distances of many kilometers. The mechanism by which trees across large areas coordinate their masting is not fully understood. Temperature signals, particularly the difference in summer temperatures between years, appear to be important. A summer that is warmer than the previous summer tends to trigger masting in beech. Because all trees in a region experience the same temperature signal, they tend to mast in the same years. The advantage of masting is thought to be predator saturation. By producing nuts rarely but in enormous quantities, the trees ensure that seed predators cannot consume all the available seeds in a mast year. Some seeds escape and germinate. In lean years, the seed predator populations decline for lack of food, which reduces predation pressure in the next mast year. The forest floor after a mast year is a transformed place. The following spring, seedlings germinate in enormous numbers from the uneaten seeds. The seedling density can be extraordinarily high. Most of these seedlings will die within months. Competition for light, water, and nutrients in the shade of the canopy is intense. Only the few seedlings that establish on a suitable microsite, perhaps a small gap in the leaf litter, a piece of decaying wood, a spot where a canopy gap allows little more light, have a chance of surviving their first year. Of those that survive the first year, only a small fraction will still be alive a decade later. Of those, only a tiny fraction will ever reach the canopy. The life of a tree is in its beginning a story of enormous odds against survival. A single oak may produce hundreds of thousands of acorns in its lifetime, and need only 2 of them to reach reproductive maturity to replace itself in the forest. The relationship between deep forest and the rain that falls on it extends beyond simple water absorption. The forest generates its own local humidity through transpiration. On a still day in a dense forest, the air between the trees has higher humidity than the air above the canopy, or in adjacent open areas. This elevated humidity affects many processes. Decomposition rates of organic matter are higher in moist air. The activity of soil organisms is greater. The germination of fungal spores is favoured. The survival of small moisture-dependent animals is enhanced. The high humidity within the forest creates a buffer against the drying effects of warm weather. In a drought that would dry out an open meadow, the forest floor may remain moist for weeks longer because of the combination of shading and elevated humidity within the canopy. Dew formation on forest surfaces at night is a significant source of moisture in some forest types. As surfaces radiate heat to the night sky, they cool below the dew point of the surrounding air, and water vapor condenses on them. Leaves, spiderwebs, soil surfaces, and rocks accumulate thin films of liquid water. This dew can be absorbed by plants, drunk by small animals, and can rehydrate dried moss and lichen tissue. In coastal forests that experience frequent fog, the deposition of water from fog onto leaf surfaces, called fog drip, can be a substantial component of the total water input to the ecosystem. Coastal redwood forests in California receive significant amounts of water from fog drip that does not register as rainfall. The trees intercept the fog with their foliage, and the collected water drips to the forest floor where it is absorbed by roots. Studies have shown that some redwood trees derive a significant fraction of their total annual water budget from fog. The redwood forests of the North American Pacific coast are among the most impressive deep forests on Earth. coast redwoods grow to the greatest height of any tree species, with the tallest measured individuals exceeding 115 meters. The diameter of the largest individuals exceeds 9 meters. These trees can live for more than 2,000 years. The mass of a very large redwood individual is extraordinary. Its trunk contains hundreds of cubic meters of dense, tannin-rich wood. The tannins and other chemical compounds in redwood wood give it remarkable resistance to decay and insect damage. A fallen redwood log may persist for hundreds of years on the forest floor, slowly decaying while providing habitat and slowly releasing its stored carbon into the soil. The old-growth redwood ecosystem supports an extraordinary diversity of organisms that are adapted to the specific conditions created by these ancient trees. Some species of moss and liverwort in the redwood forest grow only in the deep shade of old-growth canopies and are found nowhere else. Certain fern species depend on the specific chemistry of decaying redwood logs. The marbled murrelet, a seabird, flies inland to nest on the wide horizontal branches of old-growth trees, including redwoods. It does not build a nest but lays its egg directly on a large moss-covered branch. The loss of old-growth redwood forest has been a major factor in the decline of this bird. There is no substitute for a branch on a tree that is several hundred years old. The study of forest ecology has been transformed in recent decades by new technologies. LIDAR, which stands for light detection and ranging, uses laser pulses fired from aircraft to measure the three-dimensional structure of forest canopies at high resolution. A LIDAR scan of a forest can reveal the height, density, and three-dimensional arrangement of forest structure across thousands of hectares in a single flight. This allows ecologists to quantify forest structural complexity at a scale previously impossible. Satellite-based sensors can track changes in forest cover and canopy greenness across the entire Earth. Environmental DNA techniques allow scientists to detect the presence of organisms from the traces of DNA they leave in soil, water, or air samples without ever directly observing the organism. These methods are revealing new dimensions of forest biodiversity and function. The deep forest is also studied through its sounds. Passive acoustic monitoring using autonomous recording units placed throughout forest landscapes captures the acoustic activity of the entire community over months or years. Machine learning algorithms are then applied to these recordings to identify the calls of individual species. By tracking changes in the frequency and timing of calls across seasons and years, researchers can monitor population trends in birds, frogs, and insects without ever entering the forest to count animals directly. The acoustic approach is particularly valuable in remote and inaccessible forests where direct observation is difficult. One of the quieter phenomena in the deep forest is the process of facilitation, in which one species creates conditions that enable another species to survive. Nurse plants are a classic example. A spiny shrub growing in a harsh, open environment can create a shaded, sheltered microclimate beneath its canopy in which seedlings of slower-growing trees can establish. The seedlings are protected from browsing animals by the shrub's spines. They grow slowly in the shelter of the shrub. Eventually they overtop the shrub and shade it out. The tree has used the shrub as a stepping stone and then eliminated its facilitator. This kind of successional facilitation followed by competitive exclusion is common in many ecosystems and is a reminder that ecological relationships are rarely static. Succession in forests is a gradual and long-term process. After a major disturbance, such as a wildfire or windstorm that opens a large area, the process of vegetation recovery follows a broadly predictable sequence. pioneer species, which grow quickly in open disturbed conditions, establish first. These are typically plants with small wind-dispersed seeds, fast growth, and tolerance of high light and variable moisture. Over years, they modify the environment by adding organic matter to the soil, creating shade and reducing wind. Mid-successional species which can establish in the partial shade created by the pioneers follow. Eventually, the slow-growing, shade-tolerant species that characterize the matured deep forest begin to appear. The whole process from bare ground to mature deep forest can take centuries or even millennia, depending on the forest type and the severity of the original disturbance. The concept of a climax community, the idea that succession leads to a stable endpoint, has been modified by decades of ecological research. Real forests are rarely, if ever, in a static equilibrium state. Gap dynamics ensure that small-scale disturbances continuously reset patches of the forest at earlier successional stages. Large-scale disturbances such as hurricanes, ice storms, or insect outbreaks can reset much larger areas. Climate change is shifting the competitive balance among species. Invasive species are altering the species composition of forests. The forest is always in motion. The idea of a stable or climax is better understood as a long-term average tendency rather than as an achievable static endpoint. The diversity of forests around the world reflects the diversity of climates, soils, and evolutionary histories that different regions have experienced. The temperate rainforests of southern Chile contain tree species found nowhere else on Earth, relics of a lineage that was widespread during the Cretaceous. The dry forests of Madagascar support baobab trees with enormously swollen trunks that store water for the dry season. The montane cloud forests of Central America exist in a narrow band of altitude where persistent fog keeps the trees perpetually moist and encrusted with epiphytes. Each of these forest types is the result of a specific combination of climate and geological history acting on a particular flora over millions of years. The deep forest near rivers is sometimes called riparian forest. Riparian forests are often among the most productive and biodiverse forest types because of the reliable water supply and the periodic deposition of nutrients by floods. The structure of riparian forest is shaped by disturbance. Floods knock down trees and deposit debris. Channels shift over decades. New land is created on the inside of bends and old land is eroded on the outside. The trees of riparian forests include species tolerant of waterlogging, and also those that can grow rapidly to recolonize disturbed areas. Willow and alder are common in riparian zones around the world. Their roots stabilize stream banks. Their leaves fall into the water and become the primary food source for the invertebrate communities that in turn feed the fish. The science of technology, the timing of biological events, reveals how intricately the different components of a forest are synchronized. The sequence of events in a temperate forest in spring, from the first insect activity to the emergence of migrant birds, to the burst of tree buds, to the opening of the first flowers, to the hatching of caterpillars, follows a schedule shaped by millions of years of coevolution. The caterpillars need to hatch when the leaves are young and tender. The birds need to arrive when the caterpillars are abundant. The trees need to leaf out when temperatures reliably support photosynthesis, but before late frosts damage the new tissue. Each species has evolved its own phenological schedule, and these schedules are coordinated with each other in ways that have been refined over enormous spans of time. When phenological schedules become mismatched, ecological consequences follow. Research in temperate forests has documented that in some regions, the caterpillars that feed on oak leaves have been hatching earlier in spring in response to warmer temperatures. The migrant birds that feed on these caterpillars, such as pied flycatchers in Europe, have also been arriving earlier, but not early enough to fully track the earlier caterpillar peak. When birds arrive after the peak caterpillar abundance, their chicks receive less food at critical developmental stages. Breeding success declines, population sizes fall. This kind of phenological mismatch is one of the subtler ecological consequences of climate change, and is difficult to detect without long-term monitoring data spanning many decades. The deep forest at its most undisturbed offers a kind of stillness that is qualitatively different from the stillness of an empty room or a bare field. It is a stillness full of process. Beneath the surface of every log, every stone, every patch of soil, organisms are carrying out the chemical work of life. Molecules are being built and broken down. Electrons are being transferred. Carbon is being cycled, nitrogen is being transformed, water is being moved. All of this is happening continuously and simultaneously across millions of interactions that collectively maintain the forest as a functioning system. The silence of the deep forest is not an absence of activity. It is the sound of activity so distributed and so fine-grained that it falls below the threshold of human hearing. And now, in this late hour, with all that quiet science arrayed around you, the forest extends its patience to you. The old trees have waited through many nights and many seasons. The fungal threads have extended through decades of soil. The mosses have grown their slow millimeters through years of gentle rain. All of that patience is in the air you breathe right now. The deep forest does not ask you to understand it fully. It only asks you to be still for a while, and in that stillness perhaps something of its quiet order passes into you, and you can rest now. Let the sounds of the deep forest settle around you, the slow creak of a branch high overhead, the soft drip of water from moss-covered stone, the silence beneath the silence. The forest is very old and very patient. It does not hurry. It will be here when morning comes, and so will you.